Hello everyone, welcome to our channel MedVeda, where you seek a new learning experience with me. Today our topic is Erythropoiesis, which is continuation of last video. If you gonna check it, please go and watch. The link was provided in description. Now come to topic. What is Erythropoiesis? It is the process of origin, development and maturation of erythrocytes. The main subtopics that we have to learn in this chapter are site, stages and regulation. Firstly, site. Here we have to know that from fetal life to growing ages, the production of RBC takes place in various sites and by various organs. For example, in early weeks of embryonic life, that is from 1 to 3 months, RBCs was produced by yolk sac. Here the RBCs are nucleated. And in middle trimester, that means from 4 to 6 months, liver plays a vital role, but reasonably also by spleen and lymph nodes. And during last trimester, that from 7 to 9 months, and also after birth up to 5 years of old, in this stage all bones produces RBCs. After 5 years, that is from 6 years up to 20 years, long bones such as femur, humerus and membranous bones such as vertebrae, sternum, ribs and ilia helps in production of RBC. Beyond 20 years, only distal part of long bones and membranous bones helps to produce the RBCs. And next subtopic is stages. Total this process takes nearly 7 days. Starting 5 days known as differentiation phase, which is from proerythroblast to reticulocyte, and remaining 2 days known as maturation phase, that is from reticulocyte to transform to mature RBC. Okay. Now we go through genesis of RBC step by step. MHSC is multipotent hematopoietic stem cell. MHSC is a basic cell of production of entire formed cells in our body. It is divided into myeloid and lymphoid cells, which is intermediate stage cells, which very much like multipotent hematopoietic stem cells. This is also known as colony forming units. This lymphoid cell is divided to form B and T lymphocytes, whereas myeloid cell is precursor to RBC, WBC and platelets. Now we go through RBC genesis. With help of some factors like growth inducers and differentiation inducers, these myeloid cells develop into proerythroblast. It is the first stage of erythropoiesis. It is also the first cell identified as RBC characters with presence of nucleus and large size of cell and absence of hemoglobin. By multiple division of proerythroblast, it develops into basophil erythroblast. This basophil erythroblast known as first generation of cell because it's stained with basic dyes. And this stage consists of more mRNA, so it shows active mitosis. Due to its active mitosis nature, develop into polychromatophil erythroblast, which is the third stage of erythropoiesis. In this stage, we observe first hemoglobin appearance and reduction in mitosis compared to previous stage, and it later developed into fourth stage known as orthochromatic erythroblast. Here we see increase in hemoglobin and nucleus starts condensed and extruded from the cell. That means no mitosis takes place. After extrusion of nucleus, it transformed to reticulocyte stage, which is fifth stage. This reticulocyte is also known as young RBC with no nucleus but cytoplasm contain endoplasmic reticulum, basophil, Golgi apertures and mitochondria. This reticulocyte transform into bloodstream by squeezing through pores of sinusoidal capillaries. This process known as diapedesis. it was important term, diaperesis. After reaching bloodstream, it starts to mature and form mature RBC erythrocyte within 2 days. So this phase named as maturation phase. Now finally we got mature RBC, also known as erythrocyte. The main characteristics that observed in this RBC development is reduction in cell size, second, slowly disappearance of nucleus, and lastly, increases of hemoglobin. Now we have to know lifespan of RBC, which is 120 days. After this, destruction takes place in spleen, which we discuss detail in further videos. Okay. Next, the shape is biconcave with diameter 7.5 micrometer and thickness 2.5 micrometer at thick end and 1 micrometer or less at center. Next, hemoglobin content varies with gender. For male of 15 grams per 100 ml and for female of 14 grams per 100 ml. Now we go through detailed structure of RBC. RBC with non-nucleated, absence of organelles and also mitosis. We already discussed shape and hemoglobin content, and the next character is presence of blood grouping antigens, which helpful in finding of blood groups. This RBC structure had minimum tension. Due to this, it easily folded through narrow capillaries. Because of biconcavity of RBC structure shows large surface area and quick exchange of gases take place. Next, the main structures present in the RBC are plasma membrane, membranous proteins such as spectrin, ankyrin, protein 4.1, 4.2, glycophorin of A and C, CD47, band 3, stomatin, and next cytoplasm and hemoglobin. The importance of hemoglobin we'll discuss in functions of RBC. Okay. Here the count. Male had 5 to 5.5 million per microliter, whereas female had 4.5 to 5 million per microliter of RBCs. Now come to functions of RBC. The major function is transport oxygen from lungs to the tissues, where it transport along with hemoglobin. The note is 4 molecules of oxygen bind to 1 molecule of hemoglobin. Besides the function, it is responsible for acid-base buffering power. For example, if your body contains large amount of carbonic anhydrase enzyme, which catalyze reversible reaction of carbon dioxide and water to carbonic acid, the rapidity increase of H2CO3 makes possible for the water of blood to transport carbon dioxide in the form of bicarbonate ion to lungs, where it transforms to carbon dioxide in lungs and expel out as waste product, where hemoglobin bind to H plus and act as excellent acid-base buffer. Thanking you. The regulation of erythropoiesis cover in next video. So please tune to our channel and support the channel. Please share, like and subscribe.